بسم الله والصلاة والسلام على رسول الله. السلام عليكم ورحمة الله وبركاته. بداية تقبل الله طاعتكم، وأسأل الله أن يعيننا على حسن الصيام والقيام فيما تبقى من أيام من هذا الشهر الفضيل. I am Ameen Ammar, and in this video I'll be explaining the mechanism of actions of the hormones insulin and glucagon and how they regulate the enzymes in our body in order to maintain the blood glucose within the normal limit. So starting with a quick overview, both insulin and glucagon are peptide hormones, and they work by either adding or removing a phosphate group from the subsequent enzymes, and this addition or removal of phosphate group will result in either activation or inactivation of that enzyme. We will start by discussing the signaling pathway of insulin, and then we will go to glucagon. Now, as mentioned, insulin and glucagon are peptide hormones, which means that their receptors are present on the cell membrane of the cells. Starting with the insulin receptor, it's a tyrosine kinase coupled receptor. It consists of 4 subunits, 2 extracellular alpha subunits, which are the insulin binding site, and 2 transmembrane beta subunits, which are the ones that are connected to the inside of the cell and coupled to the tyrosine kinase enzyme. The story starts with insulin coming and binding to the alpha subunits. This will result in dimerization of the receptor, activation of the tyrosine kinase enzyme, phosphorylation of the intracellular tyrosine residue, and this phosphorylation will activate a subsequent cascade of reactions, resulting in many molecules in the intermediate, and the pathway will end up with the formation of an enzyme or activation of an enzyme that is called a phosphatase enzyme. Phosphatase enzyme, from its name, is an enzyme that removes a phosphate group from other enzymes, and this removal of a phosphate group will result in either activation or inactivation of that enzyme. Going to glucagon, again, its receptor is present on the cell membrane, but the glucagon receptor is a G protein coupled receptor. The story starts with glucagon binding to its binding site on the receptor. This will trigger the activation of the G protein, which will go and activate adenylyl cyclase enzyme, which will result in the formation of cyclic AMP. Subsequent reactions will occur, resulting in the activation of an enzyme that is called protein kinase A enzyme. From its name, again, it's a kinase enzyme, which means that it will add a phosphate group to the subsequent enzyme, resulting in either activation or inactivation of that enzyme. In contrast to the insulin signaling pathway, the end result was phosphatase enzyme, which will remove a phosphate group from the subsequent enzyme. Now we will add a few more points for each of the hormones, starting with insulin. It's secreted in cases of hyperglycemia from the beta cells of the pancreas. We have mentioned that it causes dephosphorylation, and it activates all the pathways that will result in a decrease of glucose in the blood, either by degradation of glucose or by the conversion of glucose into the glycogen or the lipid forms, storage forms. For the glucagon, it's released, or the main trigger for its release is a hypoglycemic state released from the alpha cells of the pancreas. It promotes phosphorylation of enzymes by the protein kinase A enzyme, and it will work to increase the glucose in the blood either by breaking down the storage forms or by synthesis of new glucose molecules. So now we have glucose that is being either degraded or synthesized, converted to its storage forms, or the storage forms being degraded to produce glucose. And by now we can understand that the conversion of glucose into any other molecules is stimulated by insulin, because insulin is activated in cases of high blood glucose levels, and the production of glucose either by degradation of its storage forms or the synthesis of new glucose molecules is stimulated by glucagon hormone. because glucagon is released upon sensing a low blood glucose levels. What we need to know from this is that for each of the processes, we have a rate limiting enzyme that controls this process. And this enzyme can be either phosphorylated or dephosphorylated by insulin and glucagon. And this phosphorylation or dephosphorylation will result in either activation or inactivation of that specific enzyme. Now, how can we understand or how can we know if that enzyme, if the phosphorylation or the dephosphorylation of that enzyme will result in its activation or inactivation? We can know this by just understanding what is the end product of this enzyme or what pathway is this enzyme controlling. Taking the glycogen pathway as an example, we have the glycogen synthase enzyme controlling the conversion of glucose into glycogen, and we have the glycogen phosphorylase controlling the degradation of glycogen into glucose molecules. If we ask ourselves whether this enzyme will be active phosphorylase enzyme or inactive upon phosphorylation. Now, to answer this question, we have to know, or we have to ask ourselves again, is this enzyme acting in a way that will result in decreasing the blood glucose levels, or it will increase the blood glucose levels? Now, we said that this enzyme controls the degradation of the glycogen store to produce glucose. So it will increase the glucose in our blood. So this enzyme will be active under the influence of glucagon and not insulin. And we have discussed that glucagon effect is influenced or mediated by the intracellular enzyme called protein kinase A. And we say that protein kinase will add a phosphate group to enzymes. So if this enzyme should be working in or under the influence of glucagon, and glucagon works by adding phosphate groups to different enzymes, this means that this enzyme will be active in its phosphorylated form and will be inactive in its dephosphorylated form. And the same applies for all of the enzymes. Like, for example, when talking about the glucose metabolism in the middle, the rate limiting step for the degradation of glucose is the phosphofructokinase 1 enzyme. This enzyme functions to degrade glucose to acetyl-CoA and different molecules to undergo the TCA cycle and produce energy. So it will reduce the glucose levels in the blood. So it will be activated under the influence of insulin and not glucagon. And we know that insulin effect is influenced or mediated by the intracellular enzyme called phosphatase. So the active form of this enzyme should be the dephosphorylated form, and the inactive form of this enzyme will be the phosphorylated one. The same concept applies for all the other enzymes, for the lipase, acetyl-CoA carboxylase, the fructose bisphosphatase 2, and the glycogen synthase. Let's see. Glycogenesis versus glycogenolysis. Glycogen synthase, which is the enzyme that will convert glucose into glycogen, it is active when it is dephosphorylated under the influence of insulin. The opposite goes for the glycogen phosphorylase, which will be active when it is phosphorylated under the influence of glucagon. This figure summarizes both enzymes and their status in the presence of insulin or in the presence of glucagon. For the glycogen synthase, it will be activated by dephosphorylation under the influence of the phosphatase enzyme triggered by insulin, and it will be inhibited, on the other hand, by the protein kinase A upon phosphorylation. The opposite goes for the phosphorylase enzyme. It will be active under the influence of the glucagon protein kinase A pathway, and it will be inhibited under the influence of phosphatase enzyme. Second is the glycolysis or gluconeogenesis. For the glycolysis, the rate limiting enzyme is the phosphofructokinase 1. It will be active upon dephosphorylation under the influence of insulin, and it will be inactive in the presence of glucagon, and the inactive form is its phosphorylated form. The gluconeogenesis, the rate limiting enzyme is the fructose bisphosphatase 2. It will be active in its phosphorylated form under the influence of glucagon. This figure summarizes what I have mentioned, and this figure also summarizes the same but with a little bit of details. Moving to the lipid metabolism, the lipogenesis will be activated under the influence of insulin. The rate limiting enzyme is acetyl-CoA carboxylase. It will be active in its dephosphorylated form under the influence of insulin. And for the lipolysis, it is the hormone-sensitive lipase enzyme, which will be active in the phosphorylated form under the influence of glucagon. This figure summarizes what I have just said for the production or degradation of the lipid molecules from glucose. Under the influence of glucagon or epinephrine, protein kinase A will be active. Phosphorylation of the acetyl-CoA enzyme will result in its inactivation. Here it is phosphorylated, inactive, and this will result in lipolysis because the other hormone, which is the lipase enzyme, will be active upon phosphorylation. And the opposite goes in the presence of insulin, which will trigger the phosphatase enzyme, causing dephosphorylation of the acetyl-CoA carboxylase, resulting in its activation and formation of lipid molecules. This is a summary of what we have explained in the video. Another figure that summarizes the functions of insulin and glucagon. Another table that summarizes the effects of insulin on metabolism. And that's it for this video. Thank you for watching, and I hope this video makes understanding this concept much more easier for you. بحب نختم الفيديو بآية: أعوذ بالله من الشيطان الرجيم قالوا سبحانك لا علم لنا إلا ما علمتنا إنك أنت العليم الحكيم. صدق الله العظيم. لا تنسونا وأهلنا من دعائكم في هذه الأيام الفضيلة. والسلام عليكم ورحمة الله وبركاته.